grade 12 learners. Welcome to today's Life Sciences lesson. My name is Agnes Moarelo, and this lesson is brought to you by the Gauteng Department of Education in collaboration with Saibono Discovery Center. I'm going to share with you the WhatsApp number where you can write your questions. However, we don't need voice recordings there or video calls. The WhatsApp number is 079-454-9535. Let me repeat it for the last time. The WhatsApp number where you can raise your or write your questions is 074-454-9535. So for today's lesson, myself and my co-presenters, Samantha, will be focusing on difficult questions for paper 2. Right, on this screen, I have this picture just to highlight to you that you have come to the end of your marathon and you decided not to stop. So keep on pushing harder and harder. Keep on working harder and harder. I'm going to encourage you to study and study and study, because at the end of the day, you're going to enjoy the fruits of hard working, right? Now, for your final push, these are the resources that you still need to use. By the way, with your exam guideline, anything that comes out of the exam guideline is examinable. Hence, we are saying as you are doing your final push, use it. Because if you are out of exam guideline, you're going to lose the marks. You still have the past papers that you need to refer to. Make sure that you compare all those questions for all the topics. Check the format on how they were framed so that you are able to know how to tackle those questions in all the papers. You still need to use your mind the gap so that it helps you to get that 45 marks out of 150. We need all of you to pass. And then you still have the broadcast lesson where you can just enhance your learning as you do your preparation for the last or final paper. Please, from my side, study and study and study. Right, just for a quick recap on the topics for paper 2, we've got DNA code of life, which has got 27 marks. We've got meiosis, which is for 21 marks. And then we've got genetics and inheritance, which is 48 marks. Lastly, we've got evolution with 54 marks. And if you can check, most of the weightings in paper 2 comes from genetics and inheritance as well as evolution. So you really deserve those marks. Work for those marks. And I know we have just written our prelims, our paper 1 and paper 2, but don't forget the focus for today is on paper 2. Yes, most of the questions, when I looked at the paper, they seem to be very easy and simple. However, after the marking of your scripts, after the marking of your answer sheet, we have realized, we have discovered that actually some of those questions were difficult, right? Now, I'm going to start with the multiple choice question. I've just sampled those difficult questions only, those that I think, or those that we think after marking, they were a little bit problematic. So with all your papers, girls and boys, you start with multiple choice question. Now on the multiple question, what you need to do for you to get it right, make sure that you look at the, stem of the question. Then we have got 4 alternative answers. You can just hide those answers, read the stem of the question, eliminate those questions that you think they are incorrect, right? then you write the correct one. Yes, at some stages you can feel that you don't know the answer, but I'm going to encourage you not to leave any space. Attempt all questions. By the way, you've got 25% chance of guessing it right. You can guess, you can get it right. All right? This question comes from KZN Prelim Paper 2 of 2024. Right, I'm going to read the STEM so that we do it together. And yes, with some of the calculations, it depends on the question. The approach differs. Now let's read this question. It says, DNA was extracted from the cells of the cat. Remember, all organisms have got their unique DNAs. And the DNA was analyzed for its nitrogenous base composition. Just for a recap, remember, with our nitrogenous bases, yes, we know that A complements T, adenine and thymine. We've got C complementing G, cytosine and guanine, right? Now, according to the question, to the stem of the question here, it was found that 26% of the base of the guanine, it was found that 26% of the bases are guanine. 26% of the bases are guanine. Therefore, you can just use a general rule there because the question is already in percentages. I mean, the stem is already in percentages, and we need what? The percentage. So it's already in percentages. Most of the guanine are at 26%, and it was found that 26% of the bases are guanine. What percentage of the bases are thymine? We need this one, right? So, like I was saying, the approach might differ. You can have an alternative answer to get it right. So guanine is 26 and it complements the cytosine. So the two can give us 52, right? And then we are left with what out of 100? We are left with 48. And 48 can actually be our A and the T. That gives us 100%. Now let's look at the answers there. What percentage of the base are thymine? Is it 26? No. Is it 52? No, because we have looked at the general composition there. Is it 24? Yes, we have said the A is the A and the T. We were left with 48. Therefore, each will be 24. So the answer is 24 there. All right. Now let's look at the Gauteng Prelim Paper 2 of 2024. Now, the important thing also, before you can attempt your answers, what you need to do is to analyze what is given. So you are given table 1, you are given table 2. Yes, you know that this is the base triplet. That's the information that is given. Just interpret it before you answer. Human DNA sequence, these are the codons here, there are codons there. And then we are given that table 2 has got mRNA codons with what? With amino acids, right? Do not worry about the names of the amino acids because they will be given in the papers, right? Now the question says, name and describe the process in protein synthesis, in the formation of the proteins that is responsible for the formation of what? mRNA. For the fact that we need the name, it means that name is compulsory. You need to have that name. And then you describe. So description of protein synthesis, girls and boys, it must be according to exam guideline. If you miss one word or the logic is not according to, it does not flow as expected, you might lose the marks there, right? Now, the name and the description of the process. The name of the process is transcription. So it's important to write the name there. Then you describe. In your description, if a question needs you to describe, in our life sciences, we need to see the cause. We need to see the effect. We need to see the statement. We need to see the reason. Why are you saying that? Okay, so with transcription, it says, according to exam guideline, remember, you need to use the exam guideline until the last day of the exams. So the double helix DNA unwinds. This is key. You need to describe the shape of the DNA. Now the shape of the DNA is double helix. So the double helix DNA unwinds, the double stranded DNA unzips. Be careful again here, because with the logic, it can't unzip, then unwind. It must first unwind, then unzip. That's the logic. So you already have what? Your 2 marks there. Then it unzips, or you can say the weak hydrogen bonds break to form 2 separate strands. This is straight from the exam guideline. Another mark, 1 strand is used as a template. Another mark, to form what? mRNA. Using the free RNA nucleotides. Remember, we have got the nucleotides there, right? So it must use free nucleotides. Sorry, also this is key. If you miss out the free RNA nucleotides, you can lose your mark there. So hydrogen bonds break, 1 strand is used as a template to form mRNA using what? Free RNA nucleotides from the nucleoplasm, or the mRNA is complementary to the DNA. Then the mRNA is complementary to the DNA. Another mark. So the description of transcription must be according to the exam guideline, girls and boys, so that you don't lose marks. So I've noticed that most of you are losing marks here. You just say the DNA unwinds, the DNA unzips without describing the shape. So it's double helix DNA unwinding and unzipping there. Okay, right. The question continues. Remember, you've got the table there. So we're given the human DNA sequence, and then we're given the mRNA codons, the amino acid there. Okay, so give the mRNA codon that is formed from base triplet 4 and on the DNA sequence. What is the base triplet 4? We are given base triplet 4, this one. All right. So before you can even attempt those answers, help yourself. You can just get the mRNA for these codons before you can even start. So ACA will be UGU. Let's go that way. TGC, right? You need to get the TGC, the mRNA for TGC, for ACG, and also for TGT. So it will be ACA. Let's go to ACG. It will be UGC, and TGC, it will be A, C, and G. So you already have your answers. Remember, for you to attempt to get them right, give yourself answers first, so that you just confirm when you go to the answer, right? Now give the mRNA codon that is formed from base triplet number 4. 4 is UGU, right? Let's look at our answer there. UGU, correct. So that is why I'm saying the tip that I'm giving you, give your answer, give yourself answers first, then attempt. So it is UGU, it is correct. Okay, now name the amino acid coded for by base triplet 2. You go to the table. This is base triplet 2, right? We've got what? UGC. I'm sorry if you can't see that, right? Right, name the amino acid. Okay, so you go to your codons there, you identify where we find the UGC, right? And then name the amino acid coded for by the triplet C. that is the cysteine. Remember, this is UCG. So that will be our cysteine. UGC, here it is. This is our amino acid cysteine. Okay. Now 214, it says a mutation caused base triplet 1. This is base triplet 1. All right. So immediately when you have your question, you go to what is given. Right. A mutation. What is mutation? You ask yourself. It is a change in what? In the sequence of the DNA of an organism. All right. So a mutation caused base triplet 1 to change so that it looks the same as base triplet 3. Right? Now, explain what effect of mutation has on the protein formed. So what I'm going to say to you now is, yes, you can be given different diagrams, different tables. However, this is the approach you need to use to the kind of the questions that I'm dealing with now. So a mutation caused base triplet 1 to change so that it looks the same as base triplet 3. Explain what? Explain. We need cause, we need effect. Explain what effect this mutation has on the protein that is going to be formed. So the base triplet TG, you start with what is given. Now the base triplet here, we've got TGT. It will become what? To TGC. I wonder if that makes sense. Remember, a mutation caused base triplet 1, this is 1, to change so that it looks the same as triplet 3. So to show that you understand, you start with what is given. So TGT, base triplet TGT will become TGC, right? It's a mark. Therefore, what is going to happen? mRNA codon changes from ACA. Remember, we got you already gave yourself the codons for those. You already gave yourself the mRNA for that DNA sequence. So what you're going to say is ACA, this one is T. Remember, it was TGT. Now it is ACA. So the mRNA or the codon changes from ACA to what? To ACG. It's a mark. Therefore, which codes for the same amino acid? Remember, ACA, you go to your codons to locate where the ACA and the ACG are, right? This is ACA, this is ACG. Let's look at the flow again. A mutation caused base triplet 1 to change so that it looks the same as base triplet 3, these two. Explain what effect this mutation has on the protein formed for 4 marks. Look at the mark allocation again. So explain what effect of this mutation, what effect of the change in the sequence of the DNA of this will have on the protein that is going to be formed. So the base triplet TGD will become TGC. Therefore, mRNA codon changes from what? From ACA to ACG. We are not going to accept if you just say ACA changes to ACG. You need to tell us that this is the codon. You need to tell us that this is mRNA changing from what? From ACA to AC. It is very, very important. Don't just say ACA changes to ACG. What of ACA? Codon changes from ACA to ACG, which codes for what? For the same amino acid. Look at the same amino acid there. The same amino acid, which is 309. Okay? So to show that you understand, you can give right to say it codes for the same amino acid or the 309 to convince us that you know your work. The protein will not be affected. Hence, or we can say the same protein will be coded for. So this is very, very important. So I'm giving a scenario on how to approach this kind of question. You can look at the prelim paper 2 2024. You can also look for this type of questions from maybe paper 2 of 2023 or 2022. The approach should be the same. I hope that makes sense. Right? Now, prelim paper 2 of 2024, yes, this question looks easy as you look at it, but upon my key, we realized again that this was a difficult question. The diagram below shows a cell during a phase of meiosis 1. Remember, there will be a difference between meiosis 1 and meiosis 2, right? Looking at the phases. And a phenomenon that can occur during this phase of meiosis, which may lead to what? Chromosomal mutation in the chromosomes, right? The question says, at the end of meiosis, gametes are produced. Yes, that's what should happen. Explain one biological importance of meiosis. Explain one biological significance of meiosis, right? One important function of meiosis is that it overcomes the doubling effect of fertilization. I know most of you right now can get it right by halving the number of chromosomes. Okay, thus it maintains, in halving, it maintains the constant chromosome number from generation to generation. The negative way, or the other way you can say, it introduces genetic variation, yes, through what? The crossing over. When crossing over takes place, that's genetic material are exchanged. So it introduces genetic variation through crossing over and the random arrangement of chromosomes, thus increasing what? A species chance of survival. And that is for how many marks? 3 marks. So immediately when you see 3 marks, make sure that you've got your 3 points there, even if there are more, so that you can get all those marks. Right? Tabulate 2 differences between meiosis 1 and meiosis 2 for the fact that we are saying you need to tabulate. So this is the free mark that you are going to get for making sure that your table borders everything, that the headings are correct. So you get mark for that, 1 mark for the table. So in this case, we need just 2 differences. Now, 2 differences between meiosis 1 and meiosis 2. The chromosomes arrange at the equator in homologous pair. This is key. However, what is going to happen in meiosis 2? They arrange themselves at the equator of the cell individually or singly. You just can say singly. Now, what is important again when you compare the two, or when you get the differences? When you talk about the apples this side, on the left-hand side, talk about the apples on the right-hand side. You can't talk about the apples this side and talk about the bananas this side. So what do you need to do? Chromosomes are aligned at the equator in their homologous pairs, where in meiosis 1, whereas in meiosis 2, they align themselves singly or individually at the equator. Chromosome number is halved, whereas in meiosis 2, the chromosome number remains the same. And whole chromosome move to opposite poles. Talk about the whole chromosome. Talk about homologous pairs of chromosomes. Because if you just say chromosomes, you might lose the marks there, right? So is whole chromosomes move to the opposite poles of the cell due to what? Contraction of the spindle fibers. Then, or you can say homologous pairs of chromosomes separate to opposite poles. Whereas in meiosis 2, what is happening? We talk about the chromatids or daughter chromosomes. What happens? They move to the opposite poles. 2 cells form at the end of cell division in meiosis 1, whereas we are expecting at the end of meiosis 2 to have what? Formation of 4 cells. Now, in meiosis 1, crossing over takes place, and then whereas in meiosis 2, crossing over does not take place. So, girls and boys, we needed 2, but it's important to know all of those because we can extend to 4 or all of them. So, those are the differences between meiosis 1 and meiosis 2. Draw a label diagram of this cell in the face before the one was shown above. So we displayed phase of meiosis. Now we need to know before that one, what was the diagram before that one. So it is important to know all the phases of meiosis, 1 and also 2. So before that phase, it is the diagram of what? Metaphase 1, right? Now, when we mark your diagrams, any diagram that we need you to draw, make sure that it has got a heading, make sure that you can label at least more than 4, or label everything so that we can just choose on what to mark, right? So this is a structure. This is the diagram before that one. Yes, we know this is metaphase. Obviously, before metaphase is anaphase 1 or anaphase 2. So that was their stage. Then before that one is metaphase 1. Now, what happens in metaphase 1? You tell yourself, we need to see what crossing over has taken place. Chromosomes are arranged at the equator in their homologous pairs, right? Then you can give any correct label there, right? This is a centromere, spindle fiber, homologous chromosome. This is a chromatid. This is a centromere. This is a cytoplasm. So you can just convince us, give us many labels there. Now, this is the way we mark any drawing that we require you or we request you to draw. So it must have, with this one, if the caption is right, you get a mark. If you have got correct representation of 2 homologous pairs, remember we talk about homologous pairs of chromosome. If we can see that they are aligned at the equator, you get a mark. And then for the shading, to show that crossing over has taken place, there was genetic variation there. For shading and for correct shading of the chromosome, you get a mark. And also for any correct labels, you get a mark. So it's easy, right? So this will happen to all your diagrams that can be given to you, okay? Right, same with Gauteng Prelim Paper 2 2024, with the same structure, with the same phase. Name and describe the phenomenon that occurred in the cell. I hope by now, because I've given you the strategies on how to answer, immediately when you name, that should be compulsory. We need that. Immediately when we say describe, we must see what the cause, we must see the effect. So this must be your receipt for all the questions. When we describe the reason statement, we need the reason, right? Name and describe the phenomenon that occurred in the cell. So what occurred is what? There was non-disjunction and the chromosomes failed to separate. If there's non-disjunction, it means chromosomes are failing to separate or have failed to separate during what? The anaphase 1. So this is what occurred in the stage. A follow-up question there. At the end of meiosis in the cell, if one gamete containing 3 chromosomes, listen to this one, if one gamete containing 3 chromosomes fertilizes a gamete containing 2 chromosomes, obviously you are going to expect what? 5 chromosomes, okay? Would this lead to Down syndrome? Ask yourself, would this lead to Down syndrome? If an individual or one gamete containing 3 chromosomes fertilizes a gamete containing 2 chromosomes, would this lead to Down syndrome? No. Why? Are we together? Give a reason. Right? According to this structure, is not, because we've got 3 and 5 and 2, they gave us 5. So it means it is not a human cell. It means it's not a human cell, because we're expecting total number of chromosomes of a human to be how many? 46, right? 44 chromosomes, then we'll be having 44 autosomes, then you'll be having a pair of what? Chromosomes. So if there's non-disjunction, we're expecting 47 chromosomes. But in that question, a gamete had 3 chromosomes, fertilizing a gamete with what? 2. Therefore, the zygote or offspring will have 5 chromosomes. Therefore, it does not have what? 47 chromosomes. And the extra chromosome is not on pair 21. Remember, if we've got non-disjunction there, and we're talking about the Down syndrome, the non-disjunction must happen where? In chromosome number 21, right? So you can be given case studies, you can be given passages to read on, you can be given any scenario, right? You can be given tables. So what is important also, we need to test to see if you can read. Now, if you're given a passage, girls and boys, you must know that some of the questions that are going to be given, answers are from the source that is given. And then also we're going to test for the content. So we can't be giving you this long passage to read without testing on what you read on, right? Okay, I'm not going to, okay, let me read it quickly. Although a rare occurrence, cases of babies being switched at birth in a hospital have made the news in the past. Since an individual DNA sequence is unique, we spoke about that one, each individual has got unique DNA with the exception of identical twins, right? DNA profiling, immediately when we talk about DNA profiling, you think of what? Relatedness of individuals. And with the DNA profiling, think of what? Think of the profiling that is presented in bars, right? DNA profiling is a reliable way of determining the parents of a given baby. Restriction fragment length polymorphism, which is RFLP, it is given though. You don't have to stress about it. Using the polymerase chain reaction PCR, right, are 2 methods used in analyzing DNA profile, okay? Below are the results of DNA profiling conducted on 3 babies and the 3 sets of parents when hospitals suspected that babies may have been switched. The hospital allocated the babies as follows, right? Baby 1 was given to couple A, baby 2 to couple C, baby 3 to couple B. This is how it is represented. Right, now I'm also going to give you a tip on how to answer this. Have your rulers, girls and boys. Right, the easiest and the quickest way, remember this is how many marks? 3 marks. You can't waste time on it. You just take a ruler, align the babies to their parents. I'm going to take this one for an example. If you use your ruler, I'm using my free hand. This for baby 1, it will be the same with couple B. You extend, right? You extend this one. You can take them 1-1, or you can just use a straight ruler to compare the similarities there if the bars are the same, okay? Now, from the text above, state 2 methods used to analyze DNA profile. We're not going to waste time on it because it's in the paragraph. This is RFLP and PCR, okay? Now, based on DNA profiling provided, which babies were correctly allocated by the hospital? Why are we saying it's baby 2? It's because we compared the bars. So you compare the bars of all the children there versus, or compare them to the bars of their parents, right? Explain why are you saying it's baby 2? Listen to the answer. You can't be saying the bars are the same. No. you must be specific to say each bar in the DNA of baby 2, each bar. Don't just say the bars are the same, that is too general. So each bar of the DNA profile of baby 2 matches with the bar of either the mom or the dad's DNA profile. So we have proved. So you compare these bars with the bars of all the parents. So each bar in the DNA profile of baby 2 matches with the bar of either the mom's or the dad's DNA profile. Or you can say 50%. How many bars are there? 1, 2, 3, 4, 5, 6. 50%, meaning 3 of the DNA bars of the baby. Talk about the bars. 3 of the DNA bars of the baby 2 matches with mom's, and 50% of 3 of those bars matches with the dad's DNA bars. So we are convinced that you know your story. Don't be general, too general. Be specific in all the answers, right? Then provide one other use of DNA profiling. Okay, we were checking at the relatedness of what? In the previous question of the switched babies, comparing them to the bars of their different, of the different couples there, the mothers and the fathers. Now, provide any other use of DNA profiling. Now we've got a lot of them, only one is needed. Please, if you happen to write 2 or 3, if we need one, make sure that you start with the one that you think is the most correct one, right? So the other use of DNA profiling, it identifies crime suspects. It does not identify crime, because most of you are losing marks to say it identifies crime. No. It's general. We identify the suspects that have done crime, or we identify the criminals. The other use of DNA profiling, it identifies a genetic disorder, right? That can be identified also when for a pregnant woman, you can also test and get, you've got freedom to test and check if your coming baby will be having genetic disorders or not. Science is a miracle. You can check that. Matching tissues for organ transplant. I can't just give you my heart. I can't just donate my heart. I can't just donate my lungs, because what should happen? We need to get a matching one first. Because if it does not match, why do I donate? So your body will reject that. So it matches tissues for organ transplant and also tracing missing persons, right? The next question, we've got prelim paper 2 of 2024 KZN. So remember, when I said you can compare the sets of past papers, you can also compare from other provinces. So the way of asking, the method of asking, and even the approach of answering those questions should be the same. So I'm not going to dwell much on this one, because I've done it to the latter when we did the first example from Gauteng province, okay? So you study the table there that is given. For 2 marks, let us check. Pepsin is a protein secreted in the stomach as an inactive pepsinogen, right? This is the table with, we are given the codons there, right? Is UUC, AAG, CUG, UGG, GAU, AUC, UGG. Oh, UGG is coming twice. It's coming twice. Okay, what does that mean? What does that tell you? Okay, let's look at the questions given. Because if you look at 4 and 7, they are the same. Is UGG, is UGG. All right, let's look at the question. Give the number of different anticodons needed to form this part of pepsin protein. The number of anticodons, right? So how many do we have? We have 7 here. So give the number of different anticodons. So you literally count those. So when you count those, we've got 1, 2, 3, 4, 5. They are 6 because we can't be repeating 4 and 7, number 7, because it's the same as 4, is UGG, UGG. So they are the same, and in this case, we need different anticodons. Therefore, we've got 6 of those, right? 6. Right, name the organelle where pepsin. Remember, it says pepsin is a protein secreted where in the stomach. Name the organelle where pepsin is synthesized, is formed in a human cell. So we are saying pepsin is formed where? In the ribosomes. Okay. Now this is the KZN prelim paper 2 of 2024. Like I gave you the approach on how to answer those questions. So I'm going to be quick on this last two. We've got a table given there. Give the amino acid coded for, so this is a table, and with reference to table 1 in question 2.1 and table 2. All right. Give the correct sequence of the tRNA base triplet that brings triosine. Where is triosine? For the synthesis of pepsin. This is AUG, right? Give the amino acid coded for by the mRNA base triplet number 3. Base triplet number 3, it is the leucine, okay? Right? And then a mutation caused. Look at these questions. They are the same, right? You just have to read the question properly so that you know how to, attempt. A mutation caused mRNA base triplet number 6 to change from UUC to AUC. Also, you need to indicate that different protein will be formed. Since anticodon AAG, that is phenylalanine, right? AAG changed to UAG. And then this is phenylalanine. I hope I called it right. Yes, I called it right. It will be replaced by isoleucine, okay, in a protein. Therefore, a different protein will be formed. So if you look at the approach, it is the same with with the questions that we started with. You go to province, all the papers. This is the way you need to approach it, girls and boys. Right? Describe the type of mutation referred in that question. It is what? Change in the sequence of what? The bases. We were told, and we have proved that. And then it is change in the sequence of bases, or you can say the nucleotides in a DNA molecule. A gene mutation will then occur, right? DBE May-June exam 2024. Also, when you attempt this type of the questions, what you need to do, you study and give yourself the answers there. All right. The diagram below represents stage during synthesis of certain protein. We have already done the formation of the protein. We have already done transcription. We know what should happen. We know mRNA at a later stage moves from the nucleus out through the nuclear pore to the cytoplasm. Okay, tRNA will have those anticodons to match them to their codons. We know all that story. Identify the organelle 3, and we say organelle 3 is what? Is the ribosome, okay? For attachment of what? Amino acid. Remember, a group of amino acid is going to form what? A protein. About 20 of them. Describe the role of molecule 1. Where is molecule 1? This is molecule 1, lysine, right? Describe the role of molecule 1 during this stage of protein synthesis. The role of molecule 1, this is lysine, it brings amino acid, it brings specific. So it should be required or it should be specific amino acids, right? According to the mRNA. Codon to the ribosomes. Give the sequence of nitrogenous bases at 2. Where is 2? Let's look at 2. This is 2, right? Give the sequence of nitrogenous bases at 2. We've got, remember this is 2, we've got GGA, it will be CCU, right? On the DNA molecule that coded for, give the sequence of nitrogenous bases on the molecule that coded for histidine. Where is histidine? I'm just about to finish now so that my co-presenter will do what? The next questions. So on the DNA molecule that code for cysteine, this is cysteine, GUA, it will be GTA, right? Again, during the synthesis of same protein, there was a change in the amino acid sequence. So use the same approach that we used in the previous ones, girls and boys, so that you get it right. So I'm going to take a break now so that my co-presenter Samantha will come and present the huge marks for genetic and inheritance and for evolution. Those questions that we thought they were easy, and you also told us the questions are easy. However, after marking, it was discovered that actually those questions were difficult. Thank you very much. See you in the next session. Enjoy.
grade 12 learners. My name is Samantha Ragunanan. I'm taking over from my colleague Agnes, who has done with you meiosis and DNA. What we're going to do right now is a very, very big section. It's genetics and inheritance. So you are supposed to be sitting there with a pen and page and writing notes and highlighting the important factors, because I'm going to highlight what is important to study. Now, if you look at the diagram behind me, you can see that this section is 48 marks. So we're not playing around here. Grade 12s, we need to be serious about this topic and need to apply ourselves. And how are we going to do this? By doing constant examples. Practice, practice, and more practice. Simply reading the notes is not good enough. It's not going to cut it. You need to go over examples. So that is what we are going to do today. Yes, the section is named Difficult Questions in Life Sciences. But once you study the notes, and once you are able to apply it, it's no longer difficult. It's basically application. So we are going to teach you strategies today of how to apply your knowledge to these questions. So when we're done, it's not going to be difficult anymore. So let's look at what our workload is like. So genetics terminology, you must study your terminology in genetics. Mendel's principles of inheritance, you need to know those off by heart. I'm touching on those aspects which I'm not going to be able to cover today. Sex-linked inheritance, very, very important. Blood groups, important. Genetic lineages and pedigree diagrams. This is a common exam question. But if we move on, your paper is divided into section A and section B. In section A, you have what is called multiple choice questions. Now, when it comes to multiple choice questions, you can't just go there and guess which one. What I like to do is I like to look at the question, and I think of the answer in my mind. I don't look at the options. Because what I find is that when I look at A, B, C, and D, the way the paper is designed, it actually confuses me a little bit. I don't know about you, but it does confuse me a little bit. So what I do is I actually try and guess the answer in my mind. I write it on the side, and then I go to the options. So that is a good strategy, because sometimes when the examiners are setting the papers, the options are very, very similar. And this can sort of swerve our decision in another direction, which might be wrong. So this is basically how you would answer a MCQ question. You read the question while covering the answers. Think of the correct answer and look for your answer. Write the answer down. But now what happens if you read the question and you don't know the answer? It happens, okay? So what you need to do is, the most important thing, do not leave anything blank. Never ever leave blank spaces. Because as it says here in the bottom, even if you don't know the answer, guess one. I know this is the first time ever a life science teacher is telling you to guess, but I'm saying to you, guess one if you don't know. Because you have 25% chance of being right. So something is better than nothing. Okay, so what we're going to do when we see multiple choice questions, we're going to think of the answer first, and then we're going to find the option. And when we don't do that, we will look at what is closest. We will look at the options and we'll see which one is the most viable option. Okay, let's look at an example of your Gauteng Prelim 2024. All of you are familiar with this paper, okay, because all of you have written this in the month of September. So it says here, now, very, very important, you need to read the stem of the question. Don't look at the answers and say, oh, I know what's going on. You need to read the question so you understand what is the context of the question, okay? So it says here, a certain disorder is caused by the dominant allele F. So we know that the dominant will mask the recessive. Yes or no? Yes. So the following combination should be considered. Now, already, I know here I'm going to have to do a genetic cross. So I'm going to have to work out all of these options, and I'm going to have to look for where I don't see a capital F, because that is my dominant allele. And that's the one with the disorder, okay? So I'm basically looking for a homozygous recessive. I'm looking for a small f and a small f. I already know that just by looking at the question. So I can already eliminate. If I look at this, option 2, it has 2 capital F's. That is not going to be viable. If I look at this one, it has 2 capital F's. It's not going to be viable, because whichever way I make the genetic cross, I'm going to have a capital F in the answer. So what I am going to do is I'm going to work out in the exam, because I am doing this in the interest of time, but in the exam, you must work out every single option and then you come to the conclusion of your answer. But for the sake of explanation, I will do the two. So we have F and capital F and small f. So then we're going to make our genetic cross. Genetic crosses are so simple. They are so simple. All I have to do is take the one letter, combine it with the other letter. Simple. There is nothing difficult about this. So those of you who have this mountain in your mind saying that it's difficult, it's really not. Let's go. So we have small f, capital F. I just write the capital F down first and then the small f. Then, so we're doing now number 2, okay? So then we have capital F, small f, capital F, small f. And then I go to the second allele, so it's small f, small f. Can you see here, already I have a recessive, homozygous recessive. Is there a capital F there? No, there isn't. That means this one doesn't have that disorder. And then if I go to the next one, small f, and so on. So I can already see that this is going to be one of my options. So let's look at the third one, because we've already eliminated the others. So we've got capital F, small f, capital F, small f. So now I'm going to do my genetic cross. So there's capital F, capital F. Can you see already? I already know that this one is going to have the disorder, okay? And then we have capital F, and then we have capital F, small f, there we go. And we got small f. So already from these two options, I can see that I have got homozygous recessive. That means there's no capital F in here. That means this certain disorder is not going to be in these two options. So therefore, as you might have guessed by now, my answer would be, okay, let me just go back. My answer would be A, okay? So my answer is going to be A, and we know why right now. So let's continue. We're going to go to KZN Prelim 2024. Now, if you look at this diagram, it is a very familiar diagram, okay? It comes out very often. So we're going to get straight to it. We already know from our work in class, because this is a revision lesson, that we have the affected, which is in bold, and we have the unaffected, which is not colored, as you can see there, okay? And the female is depicted by a round circle, and round circle synonymous, yes, I know, okay? And then you got the square. The square is the male. So what we need to do is, just by looking, yes, we're going to get to the questions in a while, but what we're going to do right now is we're going to look at the diagram itself, and we're going to try and make sense of the diagram. Remember, when we look at the question, sometimes it can make us a little distracted. So I want to first make sense of the question. So when I look at the question, I know that I'm dealing with the inheritance of fur color in rabbits. So I know that there's brown fur, and I know that there is white fur. Okay, so we've got brown rabbits and we've got white rabbits. But we already can see, if I count the number of brown fur, 1, 2, 3, 4, 5, 6, 7, 8, I already know brown fur is dominant. It is the dominant allele, because most of the parents and the offspring is brown fur. And then I know that obviously what the clear one is the unaffected, which is the recessive allele, and we know that that would be the white fur. Okay, and let's go to the questions now and see what we can make of it. We know that you have the parents, there is a female, there is the male, and there is a they mate, and then the offspring. So that would be the first generation. We know that's the second generation, the third. This is all work that we've done in class. So we must apply our knowledge. Okay, we're not just going to in the exam give the facts we got in the class. We are applying the knowledge. That is why it's important that we do past year papers. Okay, so let's see 2.4.1. The question says, name the phenotype. Remember, phenotype is physical features. Pheno for photo. So we want to see what is the visible characteristics that is controlled by the recessive allele. And we already mentioned that. We said it's going to be the white fur. Okay. Explain your answer in 2.4.1. So let's look at example 3 and 4. If we look at 3 and 4, those parents, both of them are brown in color. But the offspring here, look at the offspring at number 7. The offspring has white fur. What does it say? It says that each of these parents have a recessive allele. Because in order to get a white furred rabbit, we need to have small b, homozygous recessive. So that explains our answer. If we look at it, both individuals 3 and 4 have brown fur. We can see that. How do we know that? We didn't guess it from our minds. The answer is in the question. Okay? The offspring 7 has white fur, which is expressed in the phenotype of a homozygous. Remember, in order for homozygous to be reflected in the offspring, it must be both small letters. Okay? Each parent must be heterozygous. They must be heterozygous because they're brown in color. So within the genotype, there is a recessive allele, and that would make it possible for it to be carrying a recessive allele, and the offspring inherited the recessive alleles from both parents. There we go. So it's simple. So we're moving from difficult to simple. Let's go to 2.4.3. Same exam paper, same question. There's a variety of questions they can ask you. Identify the parents that support the conclusion that both parents are with brown and white fur coat belong to the same species. Now, it is very, very important that you understand what a species is. A species is when you have organisms that are able to interbreed and produce fertile offspring. So the fact that they're brown and white fur, what the question is telling us is that, yes, we got a parent that's brown, we got a parent that's got white fur, but the offspring, identify the parents support the conclusion that both rabbits with brown and white fur belong to the same species. So now what we need to do, we need to look at the question. From the question, they're saying the parents have brown and white. So this is the only one that they can be talking about, 5 and 6. It is the only one. So you need to identify that. So they're asking you to identify it, and that would be 5 and 6. Give a reason for your answer. So what is the reason? Simply because they must be the same species. Because remember, the definition of a species is that they are able to interbreed and produce fertile offspring. They're interbreeding because they produced an offspring. And how do we know this offspring is fertile? Because there we have number 11. 11 is their offspring. So the answer is so simple. It's staring you right. They're literally staring you in the face. Okay? So they say, give a reason for your answer. They are able to interbreed and they produce fertile offspring. This is a very important definition of species. It's going to also come up when we do evolution. So you must know this definition. Okay? State how many generations in the diagram. There is 1, 2, 3, and 4. So there's 4 generations in the diagram. Simple as can be. So do you see, even though the question appears complex, when we break it down into its components, we understand our genetics definitions, we understand how to read a pedigree diagram, we will be able to apply our knowledge and answer it. Okay? So let's go to the next question. Rabbit 11. So already they are focusing on rabbit 11. So now we need to go here. This is rabbit 11. Okay? Was crossed with a white fur coat rabbit. Now already I'm looking at rabbit 11. I can tell that it's a male. I can tell that it has brown fur. They say use the letters B and B to do a genetic cross to show the phenotypic ratio of the offspring. So what we're going to do here is, they say rabbit 11 was crossed with a white fur coat. The fact that it is white fur coat, it means that it is a homozygous recessive. That means it must be BB, because that's the only way that that option can be a white fur. So we're going to take the brown fur, we're going to name our phenotype. Firstly, phenotype, the physical characteristics, we're going to call it. Brown fur is crossed with white fur. And then we're going to get a mark for that. This genetic cross is always 6 marks, sometimes 7 marks, but it's a 6 marks which is yours to take. You just need to understand what is homozygous, what is heterozygous, what is dominant, what is recessive. Where are we going back to? We're going back to our definitions, our genetic terminology, our scientific terminology, okay, life sciences. And then we cross them. Now, there's a format to this. So your teacher probably taught you many methods to remember this format. But for a genetic cross, we can say, please, girls, make good friends. Not bad friends, good friends, okay? So we know that we must have P1. We know that we must have the genotype, which is the genetic makeup. We know that there must be meiosis, and meiosis must come before fertilization, or else it's going to be marked wrong. And we know that we must list the gametes. So there is a template, there is a format. And even if you don't get the others right, if you write, please girls make good friends, which is P1 genotype. Now don't go write, please girls make good friends. It is a mnemonic. It's a way for us to help remember these facts, okay? So you write down P1, you write down the genotype, you write down meiosis in the middle of it, which is gametes and fertilization. You'll get marks for it. Even if you don't get the genetic cross correct, you will get marks for it. So difficult made easy. You can get some marks, okay? But it's easy enough so that you can get full marks. So then we do the cross. The cross is fairly simple. You are going to, you're going to take your genotype and you're going to break it up into your gametes. Say you have capital B, small b, small b, small b, okay, coming from the white fur. Now, I'm not going to go into detail with this because we've done this when we did the multiple choice questions. All we do is we rewrite the letters. So we take the capital B, we combine it with the small b, the first letters of each cross, okay? And then we're going to get capital B, small b, which obviously by looking at this, I can see it's heterozygous, and I can already see that this rabbit is going to be brown. Okay, because we know that the brown is the dominant allele. Okay, and then we go capital B, small b. It's going to be the same. So now already we know we got two brown rabbits that we are discussing. And then we got small b, small b. This is going to be, what color is this rabbit going to be? Yes, it's going to be white. It's going to be a white fur rabbit, and because it's homozygous, recessive. And then we got small b, small b. And that is also going to, so we already know our ratio is going to be 2:2. But let's see if we correct. We correct. Okay, so our phenotype, so we need to list what our findings are. We need to list the phenotype, we need to list the genotype. So we have 2 brown fur, 2 white fur. And obviously we need to simplify. Just like in mathematics or in any other subject that involves statistics, we need to simplify our answer, and our phenotypic ratio is going to be 1:1. The compulsory mark. Do you see when you say an asterisk like this, it means that in order for you to get the full 7 marks, you must write P1 and F1. This must be there in order for you to get a full 7 marks. If you don't have P1, F1, you will only get 6 marks, even if you got everything else right. Because that's why we call it a compulsory mark. So that is why it's important. When we're doing a genetic cross, we must list all the steps. We can't forget anything. We can't leave anything out. So when we finish our examination, we go back and we check. We don't sit there looking around at everyone in the class. We go back, we check our answers to see that we didn't forget anything, because we are human and we make mistakes. Okay, and sometimes we also forget. So there's another way to do the genetic cross, which is the way I like to do it. I like to do it in a Punnett square. So this is a Punnett square which you've done in the class. It's a very same principle that we used before, but for the way I like to do it is like this, because then we don't forget anything. We don't match the wrong ones, and then we write the genotypes on the top, and we write the genotypes on the side, and then we get the answers. Very same setup, it's just that we put it in a Punnett square. The Punnett square is much easier to understand and to work out, but you will get marks regardless, even if you do the line method, as I've shown you before. So let's look at Gauteng Prelim 2024, and this is a question that is also based on genetics. Now, if you look at this question, it's a scientific investigation. That means they did an experiment. Now, I want you to pay attention when we do this, because it is law. The examiner, when they're setting your paper, they are told there must be a scientific investigation in the question paper. It is a requirement. So what do you need to know before you even start this question? You need to know how to establish the aim. You must know the dependent, what is an dependent variable? What is an independent variable? How do you draw graphs? Do I know how to draw a pie graph? Do I know how to draw a bar graph? You must know all of these things. Those of you who don't know how to draw graphs, you need to go to your Mind the Gap. There's a section there that teaches you how to draw graphs. Okay? And there's certain things that we look for when we mark graphs. So scientific investigation, very, very important. So let's read what they are saying here. Nala and Luke are farmers of the same species of a small fish. Nala breeds homozygous yellowfish. What does this already tell me? It tells me that this yellowfish, their parents is yellow and their grandparents are yellow. Okay? While Luke breeds homozygous blue. The same. Their parents are blue. So this fish's parents are blue and the grandparents are blue. The whole family is blue. Okay? That's what we're saying. It's like sort of like a pure breed. In an investigation to determine the dominant color in this species of fish, they allowed for the yellow fish to breed only over the blue fish over the years and collect 100 offspring each year to observe. So basically what they did, they got the blue fish and they got the yellow fish and they bred them. And what they did is every year for 5 years, they caught 100 of these. They allowed them to breed and then they took a sample of 100. And from the 100, this was the stats that they got. As you can see, it's put in a table form as is accepted. You can see the headings there. And we're going to go straight into the questions. So name the type of dominance that is shown in this diagram, in this investigation. But if we look at the investigation, I'm going to go quickly back. When they caught this fish, these 100 offspring, they found, of that 100, they found yellow. Remember, there was no yellow before. But when the blue and green crossed, then they became yellow offspring. So the highest number, no, there was yellow before. Sorry, I'm talking green. There was no green before. So the most number of offspring that they caught out of that 100 fish were colored green. So that is an intermediate phenotype. That means when the blue and green, they mated, they were yellow offspring, okay? And that is what they're asking you. What is the type of dominance? But because they were mostly green, we can call that incomplete dominance. What is incomplete dominance? When neither the yellow is dominant or the blue is dominant, but there's an intermediate color, okay? So that is incomplete dominance. Why? Because the green fish represents an intermediate phenotype. It's a blended version of the alleles of the parents. Two ways in which reliability of this investigation was ensured. When I say reliability, I always think in my mind, reliability, repeat. So I already know repeat, the investigation is going to be one of the answers. So how did they make it reliable? They collected 100 offspring, not 50 1 year, 20 the next year, 30 the next year, 100 offspring every year for 5 years. They calculated the average of the offspring and they repeated the investigation. What reliability does, these reliable factors, if another person has to go do the same experiment, they should be able to get the same results. That is the whole point of reliability. One way in which the results of the investigation were made more valid, they were made more valid because only yellow fish mated with blue fish. There was no other color. So therefore, both the parents were homozygous. That we discussed. 2 planning steps. Now these are the planning steps, okay? So the planning steps, we're going to list them very quickly. We decide on the best time to allow the mating of the fish. That means fish don't really mate all the time. There's a certain season, so they needed to decide. So maybe they kept the fish separately, and when it was mating season, they let them out of their nets and they allowed them to mate. How to safely incubate or store the offspring. So basically what they wanted to do is they need to incubate, they need to make sure that those offspring is actually kept alive, because it's going to influence our stats. They need to know what food to provide, decide which food to provide the offspring, because certain types of food can change the color of a fish. Did you know that? So the food, the diet that they followed was very important. They needed to decide on the sample size and how to record the results, and also how to decide the time and the date and the season of the sampling. They also need to get permission from animal ethics, okay? And even though you can write a whole lot, but they're only going to mark the first 2. So draw the bar graph to show the results. So when it comes to the bar graph, okay, when it comes to the bar graph, we need to know that on this side, the colors, the phenotype would obviously be on the x-axis, okay, because these figures are constantly changing. So the average number of fish is what is listed here. So the average number of fish is the dependent variable, okay, and that is how the graph is drawn. There's the 20, there's the 65, and there's the 15. Now you're going to get a mark. I'll show you the grid a little bit later, but you're going to get a mark for the caption. You're going to get a mark for the labels on either side. And also, remember, when you're drawing a bar graph, the width of the bars must be constant. You can't have one thin bar and one fat bar. All your bars must be the same thickness and width, okay? Now, we use the over 5 years here. So if you look at the heading, most learners, when they're writing the heading for this bar graph, they will write the average number of fish for each phenotype, because what most learners do is they take the label for the x-axis and they combine it with the label of the y-axis, and then they put their caption. But it wouldn't be correct. You need to read the stem of the question, because the experiment, remember, the experiment took place over 5 years. If you are going to say average number of fish for each phenotype, so what? You need to go back to the question, look at the question, look at where the experiment, the time frame, and it must be included there. That is why when you go through past year papers, you need to look at the context in which the question is asked. Okay, so there is the caption. We've discussed this. You get a mark for the caption. You get a mark for the type of bar graph that you draw, for the labels, for the scale. That means the distance on the x-axis between the different markers, like 1, 2, 3, 4, and also for the plotting of the graph. Now, if you have to draw a histogram or a line graph, you will lose marks for type of graph. Remember, histogram doesn't have spaces between it. All the bars are close by. So you're going to lose a mark because it's a wrong type of graph. Okay? And then if the labels are not corresponding, then marks will be lost for labels. It's obvious there. Okay? Now, I want to get to the section because this is a very, very important section. Evolution. 54 marks. 54 marks. When you start studying for paper 2, this is a section you should be starting with, because it has the most, it is loaded with marks. Okay? So, and the good thing is that you are watching this. So I'm telling you, they have questions that trend. You know, like how things trend on TikTok or on Twitter and all of those things. Those questions trend every year. So you need to pay attention. You need to be sitting there with your pen and your paper. Now, this I already can see. It says a population of squirrels. A population of squirrels lived in the mountains. A landslide caused the river to change direction, thereby separating the populations into 2 groups on either side of the river. The 2 populations became different species. So basically what they're saying, there was a section of land, there was a river that was running through this land where the squirrels stayed, but the river changed direction. So there's the river there. So what happened? These squirrels that were staying, they were separated. Now what you need to know for this, already I can tell you, you need to know the definition of population, and you need to know the definition of species. We said a population is when a same number of, when a group of organisms or a group of organisms of species stay in the same area at the same time. Okay, and this is going to be key to answering this question. So it says describe the process of speciation. Now speciation is when a group of organisms of a species, they develop their own characteristics, and that separates them from the rest of the group. So let's look at this. This is directly from the exam guideline. Okay, so the population of a single species of squirrels becomes separated by a geographical barrier. Do you see? It's a compulsory mark. You have to name the barrier. Okay, and then the population is split into 2. That is obvious. The population is split into 2. You can't say it's split into 2 species. It's incorrect, because if you go back to the definition of a species and a definition of a population, they differ. So make sure that you say the population is split into two populations, okay? There is no gene flow. Obviously, there's no gene flow. The one group of squirrels is on the one side, the other group, they are unable to mix. And each population was exposed to different environmental conditions. Natural selection occurred independently. That means they were exposed to different things. Maybe on the one side of the river, they were eating nuts. On the other side of the river, they were eating a different form of nuts or fruits. So this would change the structure of their teeth, such that the individuals of two populations become very different from each other genotypically and phenotypically. Even if they were to mix again, even if this river were to close up, now they are both formed a different species, they would not be able to interbreed. And the two populations now become a different species. You need to know the definition of population and species in order to answer this question. And it's a common exam question. So please, learners, you have to know this. Reproductive isolation mechanisms, we know it's breeding at different times of the year, species-specific courtship behavior, plant adaptation to different pollinators. That means plant will be, maybe one plant will be brightly colored, have huge petals. Another plant will have a large sticky stigma. So there's different ways in which plants have reproductive isolating mechanisms that will suit the pollinators that it wants to pollinate it. So this last diagram that we're going to do is on the skulls of 3 hominins, and I really want you to pay attention here because this is a common exam question. It comes out year after year. So they give you the diagram of the 3 skulls. They say, give the 2 structural features of Australopithecus, that one, and that are more similar to gorilla than to Homo sapiens. So there we have, so what they want to know is they want you to look at these 2 skulls and they want you to say, how are these 2 similar to each other? Already you can see the jaw is more prognathous. That means it's coming out. You can see the jaw here is larger than Homo sapiens. You can see that the brow ridges are more prominent. It's sort of coming out. So those are visible features, as well as the smaller cranium. You can see the cranium is visibly small. Look at how large the Homo sapien cranium is. And very, very importantly, it has a U-shape. It's more U-shaped. But if you look at Homo sapiens, it's more of a wider arc, okay? And they have prominent brow ridges. Now, 3.3.2, explain how the size of the canines in the gorilla is used as an evidence for their diet. You might not be able to see it, but the canines here are quite large. And why are they large? Because think about it, if you had to eat raw meat, you'll be chewing and chewing, you won't even be able to bite into it. But because the gorilla eats a lot of raw food, the canines are more pronounced and it's able to tear the food, okay? I'm going to move to the last question in the interest of time. And this is a common exam question, very, very quickly. Describe how the age and the location of hominin fossils support the out of Africa hypothesis. You must know this. It is a common exam question. What I need you to know is, what does this question mean? It means they are saying that all life originated, it started here in Africa. And they are saying what evidence is there to prove it. So what evidence is there? The oldest fossils of Ardipithecus, and remember this was 5 million years ago. Ardipithecus, the age of Ardipithecus was found in Africa only. Australopithecus 4 million years ago. Okay, it was only found in Africa. Homo habilis was only found in Africa. What does it mean? It means that life originated of hominid fossils originated here in Africa, because they only found these fossils here, and these are the oldest fossils. If you think about it, your great-grandmother was here first before you. It's the same principle. These oldest fossils were found here in Africa, and then the oldest fossils of Homo erectus were found in Africa and of Homo sapiens. So what does that mean? It means that they weren't only found in Africa. They were found in other parts of the world. But the oldest ones, the ones that first, if they took the fossils and they dated it, the oldest one was found here in Africa. So thus we can prove that the out of Africa hypothesis is quite a valid hypothesis, because most of the first and the oldest fossils were found in Africa. Grade 12s, I wish you well for your study sessions. Please tune in next week, as we're going to be doing more important work in the following section. Good luck.